Welcome to this explainer. Today we're diving into something absolutely wild. We're going to explore a phenomenal, totally invisible world that has literally shaped every single one of us. We're unpacking the fascinating science of how our lifelong health, our immune systems, and even our brain development are engineered from the very beginning by invisible microbial forces. It's a story of incredible biological architecture. And the crazy part? It starts before we even take our first breath. For the longest time, right, we all just kind of assumed that a newborn baby was a completely blank biological slate. A sterile, naive organism just waiting for the world to write on it. But the latest research, it completely shatters that assumption. As we're going to see today, there are profound maternal, prenatal, and immediate postnatal influences already hard at work constructing the body's defenses. A newborn isn't a blank canvas at all. It's a highly active, dynamic construction site.
Okay, so let's jump right into the first phase, life before microbes.
There's this huge misconception that when a baby cries for the very first time, its lungs are fully finished and ready to go. But mammalian lung development actually happens in distinct phases, and it continues well into postnatal life. During those early prenatal phases, like the pseudoglandular stage, the lungs are populated by embryonic macrophages. Now, don't think of these innate immune cells as defenders. Instead, think of them as tiny architects. They're releasing mediators, remodeling tissue, and physically helping build the respiratory branching structure long before the baby ever encounters a single breath of outside air.
And when we look at the gut, it's a completely different story compared to an adult. The prenatal gut completely lacks the deep crypts and Paneth cells that adults use to pump out antimicrobial peptides. So you might be wondering, how on earth does a newborn protect itself? Well, it relies on a highly specialized, universal neonatal antimicrobial peptide called CRAMP. Basically, the newborn gut is holding down the fort with a completely different temporary toolkit.
All right, moving on to our next stage, the postnatal microbial wave.
This right here is a massive paradigm shift. Think about an adult's immune system. It's super aggressive. It uses a really diverse, highly competitive microbiota to just block out pathogens. But a newborn's immune system, it's the exact opposite. It is highly tolerant, has low microbial diversity, and is in a crucial learning phase. We really need to start looking at microbes as teachers. The baby's immune system isn't broken, and it isn't just immature. It's deliberately turning down the internal alarm bells so it can safely learn from this brand new microbial curriculum without causing massive, damaging inflammation. But, what happens if that early curriculum is wrong? That brings us to dysbiosis. Dysbiosis is simply an imbalance in this critical microbial community. Because the gut and lung microbiomes are being established at the exact same time the immune system is developing, the nature and the precise timing of these interactions are absolutely critical. If this process gets disrupted, say by unnatural microbial exposure or early inflammation, it can literally set an individual on a lifelong trajectory toward disease. So how exactly does the body manage this? Let's look at training the immune system. The real hero here is the maternal link. A newborn's mucus barrier is thin and structurally incomplete, but maternal breast milk acts as a vital, highly engineered bridge. It's not just providing calories, it's supplying heavily armed bioactive factors. We're talking secretory IgA, specific growth factors, hormones, cytokines, and complex oligosaccharides. These essentially blanket the baby's immature mucosal surfaces, providing borrowed maternal immunity while the baby's own system comes online.
And while that maternal shield is up, the baby's own immune cells are going to school. The absolute superstars of this show are regulatory T cells, or Tregs. You can think of Tregs as the peacekeepers of the neonatal lung and gut. Early, healthy microbial colonization naturally induces these Tregs. Their main job is to enforce peripheral tolerance, which basically means they actively suppress the immune system from going haywire and attacking harmless environmental triggers or food proteins. They are literally the reason you can hopefully enjoy a peanut butter sandwich today without a trip to the emergency room.
Now, let's shift gears and look upward to the gut-brain connection.
1 in 100. That's a staggering number, and it represents the global prevalence of children affected by autism spectrum disorders, or ASD. Now, we know genetics play a massive role here, with heritability estimated anywhere between 70 and 90%, but genes alone just don't explain the whole picture. There's an emerging, highly compelling theory that the gut microbiota might actually be the missing link, bridging the gap between those genetic predispositions and early environmental factors in ASD.
So how does the gut talk to the brain? Well, it's a two-way superhighway.
Signals can travel straight up, direct neural cables, like the vagus nerve, or they can travel through immune and endocrine pathways, carrying cytokines and neuropeptides right into the systemic circulation. If the gut microbiota is altered or dysbiotic, it sends abnormal signals, rogue metabolites, and inflammatory cytokines straight up these pathways, and that can directly impact neuronal activity, myelination, and neurogenesis in the developing brain.
Let's break down exactly how this damage happens, step by step, using the idea of leaky barriers. Step 1. A microbial imbalance alters the tight junctions in the gut lining, and this is often signaled by a protein called zonulin. Step 2. This creates a leaky gut, allowing unwanted microbial metabolites to just slip right into the bloodstream. Step 3. This systemic inflammation travels up and actually causes a leaky blood-brain barrier. Step 4. Once inside the central nervous system, these inflammatory signals activate microglia, which are the brain's immune cells, causing neuroinflammation that can physically disrupt fetal or neonatal brain development.
Which brings us to the ultimate so what of this explainer: lifelong health impacts.
Our early environmental choices really stack up and physically built on each other. Aggravating factors like C-sections, early antibiotics, or severe neonatal viral infections like RSV disrupt that crucial early microbial colonization. This drastically decreases those peacekeeper Treg cells we talked about, which skyrockets the risk of lifelong asthma and allergies. But on the flip side, alleviating factors like breastfeeding or even maternal exposure to farming environments do the exact opposite. They promote an incredibly rich, balanced microbiome, increase Treg cells, and lay down a foundation of resilient, lifelong health.
I absolutely love this quote from the researchers because it really hits the nail on the head for our journey today. In the context of mucosal immunity of the newborn, it might be more appropriate to speak of a distinct rather than immature mucosal immune system. We seriously have to stop treating infants as just tiny, fragile adults with broken immune systems. They are entirely distinct, brilliantly adapted biological ecosystems that are currently under heavy construction, beautifully orchestrated by the very microbes they encounter.
So that leaves us with a pretty mind-bending question to wrap things up. Knowing the profound, life-altering power of those earliest microbial teachers, how did your own invisible architects, the microbes you met in the womb and in your very first days of life, shape the foundation of the body and brain you are using to watch this explainer right now? It completely changes how we view human health, doesn't it? Thanks so much for joining me on this incredible deep dive, and as always, keep asking the big questions.